All right, let's get right into it. Welcome to the Explainer. If you work in QA, QC, validation engineering, or production, you already know that a failed cleaning validation is basically the ultimate stress test for your pharmaceutical quality system. It is not just a minor hiccup, it's a massive deal that challenges the very core of your cross-contamination prevention strategy. Today, we're diving into the root cause analysis of failed cleaning validations. We're going to look at how to treat these failures not as a full-blown crisis, but as a systematic, scientific investigation. Let's jump in.
Picture this absolute nightmare scenario for a second. You're reviewing the latest lab results and boom, a swab test from a critical piece of manufacturing equipment comes back OOS, out of specification. What happens next? Well, the implications are immediate and severe. Upcoming product launches, put on hold. Patient safety, immediately at risk from cross-contamination. You literally cannot just cross your fingers, rewash the equipment, and hope it passes the second time. The reliability of your whole cleaning process is completely in question now, and you have to figure out exactly why.
Which brings us to the real cost of these cleaning failures.
When we move into the investigation phase, we really have to treat this cross-contamination risk as a high-stakes forensic mystery. And look, regulatory bodies like the FDA, the EMA, and PICS, they absolutely demand that you treat a cleaning validation failure exactly like any other major quality event. There is no sweeping this under the rug. Every single OOS swab test, any visible residue left after cleaning, or a repeated failure to limit residue has to be thoroughly investigated using hard, scientific data. The expectation is rigor and evidence, plain and simple.
And that means we have to look way beyond simple operator error.
This is a huge pivot point for any investigation. We need QA and engineering to dig a lot deeper. Look, I get it. It's incredibly tempting to just say, it's operator error, assume someone didn't follow the SOP, and just repeat the cleaning process. But guys, that is just a Band-Aid fix. The right approach is a genuine, scientific root cause investigation. Because cleaning validation rarely fails for just one isolated reason. It's almost always a combination of factors. Our goal isn't just to find out where the residue is hiding, but to systematically deconstruct why the process failed to remove it in the first place.
So let's look at the actual product and equipment cause.
Let's talk about the usual suspects in our mystery, the product chemistry itself and hidden machinery flaws. Honestly, some products are just inherently harder to clean because of their residue characteristics. We're talking about things with excessive amounts of sugar, thermal degradation residues, hygroscopic materials, or those really tricky hydrophobic formulations. Think about it. If you're making a controlled-release tablet with hydrophobic polymers, that residue is going to be incredibly stubborn compared to an immediate-release product made in the exact same equipment. You absolutely must understand the chemical makeup of your residue to figure out why your standard cleaning was totally ineffective.
But hey, it's not just the product, right? It's the environment. Even a flawless cleaning procedure is going to fail on poorly designed machinery. We have to examine the hidden crime scenes of pharmaceutical manufacturing. We're looking for things like dead legs, microscopic cracks, faulty gaskets, bad welds, rough inside surfaces, and old seals. Sometimes the residue is literally just building up in unreachable areas of your CIP systems. When investigations are done right, teams often have to use endoscopies or totally tear down the equipment. You would be shocked how often stubborn residue is just hiding out under unlisted removable parts.
This naturally leads us to the sinner's circle evaluation.
So, we have our suspects, but how do we actually evaluate the scene? We use this primary forensic framework called the sinner's circle. It's essentially a pie chart cut into 4 equal slices: time, temperature, chemical activity, and mechanical activity. These 4 factors completely dictate the effectiveness of any cleaning process, and the whole thing is a delicate balancing act. If one of these drops, let's say your chemical activity is low because of a diluted detergent, you have to bump up another one, like mechanical scrubbing or time, to get the same overall efficiency. When a failure happens, it means this circle was broken somewhere.
To actually build your investigation, you need to use an essential evidence matrix. You have to rigorously compare your actual cleaning parameters against your validated baseline. Ask yourself the hard questions. For time, was the validated duration actually maintained? For temperature, was the cleaning solution within the approved range? For chemical, was the detergent prepared correctly based on that specific residue chemistry we just talked about? And for mechanical, was the CIP spray pressure or the manual scrubbing truly adequate? Pinning down the objective evidence for these questions is exactly how you crack the case.
Now, let's talk about avoiding common investigation mistakes.
As you gather all this evidence, you have to be so incredibly careful.
I really can't warn you enough against these common pitfalls. Number 1. Repeating a cleaning cycle without investigating. If you do that, you've learned absolutely nothing. Number 2. Assuming a lab error. If you just instantly blame the lab for an OOS swab, you are ignoring a massive red flag. Other fatal mistakes include closing off the investigation before you actually verify the root cause, implementing CAPAs that aren't even valid, and ignoring worst-case product assumptions. Brushing off a failure leaves your entire production line wide open for a repeat disaster.
So, how do we use this to improve our future programs?
Here's how we fix it and actually evolve the system. The data you get from a failed validation is basically free intelligence to bulletproof your future cleaning programs. First, you need to evaluate cleanability right during the qualification phase. Don't wait until production. Second, always choose your detergent based on the actual residue chemistry. Third, relentlessly analyze your validation data trends. And fourth, you've got to launch cross-functional investigations. Get engineering, production, analytical, and quality all in the same room. That is how you fortify the entire process from end to end.
Which brings us to the core philosophy of this whole explainer. A cleaning validation failure should never be treated as just an isolated headache. It is a vital opportunity to assess the efficiency of your entire cleaning process, from the day you design the equipment to the moment the lab tests the swab. A truly effective investigation challenges our assumptions and relies on hard, objective evidence, not just convenient explanations.
I want to leave you with this final provocative thought. When the next out-of-specification swab result inevitably crosses your desk, what are you going to do? Are you just going to point fingers at an operator and hit rewash? Or are you going to pull out the center circle, analyze the chemistry, inspect those dead legs, and actually evolve your entire pharmaceutical quality system? The choice is entirely yours, and honestly, patient safety depends on you choosing the latter. Thanks so much for joining me for this explainer. I hope you're feeling inspired and ready to tackle your next validation challenge like a true forensic investigator. Keep questioning and keep improving.